Today we are going to we are going to discuss practical part of like sterilization practicals, microorganisms and diseases, and use of microorganisms in industry. Right. So we'll start with practicals, sterilization practicals. So in the session 3, we learn about different types of sterilizations. Mainly, we can divide methods of sterilization into two groups as physical sterilization and chemical. Under physical sterilization methods, you learn about, there are several types, right? Moist air sterilization, then boiling, pasteurization, then under dry heat, like then we learn about chemical sterilization. We learn about two chemicals, two gases that we can use to sterilize microorganisms, right? So why we need sterilization, right? It's a question now. Normally, just think we need to study about microorganisms that present in a soil sample, right? So can we do it in that particular habitat? No, we have to bring them into the laboratory. But in the laboratory, we have to provide that the conditions that they required for their growth. I mean, the moisture, sufficient amount of moisture, the suitable pH, and the necessary nutrients for their growth. Right? In the laboratory, we have to provide that similar condition to study about their, like maybe like we want to study about their structure, right? That means their shape, right? Those things. So we have to bring them into the laboratory and we have to provide that similar conditions for their growth, right? Growth and reproduction, growth, development and reproduction, right? So microorganisms can, now look at the screen, microorganisms cannot be studied in their natural habitats such as in soil, water or air. You know, microorganisms, they are ubiquitous on Earth. That means they can be found everywhere. Therefore, we need to bring them to the laboratory and provide similar conditions for their growth and reproduction, right? So the medium, right, which we provide nutrition and encourage for them is called culture medium, right? Culture medium is a nutrient material prepared for providing nutrition and encourage essential to the growth of microorganisms. So this culture medium, it includes the necessary nutrients, the sufficient amount of moisture, and the suitable pH for their growth. So you should study the definition of culture medium. In the exam, they can ask, what is culture medium? So culture medium is the nutrient material prepared for providing nutrition and encourage, right? There are two uses. It provides nutrition and encourage, which is essential for the growth of microorganism. But the next thing is, some microorganism. Now, if we take microorganisms, some are culturable. Culturable means we can grow them on laboratory, like in the laboratory, in a culture media. But not all microorganisms are culturable, right? That means we cannot grow all microorganisms in a laboratory culture media. So the microorganism who cannot grow in a culture media, in a laboratory culture media, are called non-culturable microorganisms, right? Non-culturable microorganism. And the next thing is, some microorganisms, they grow well on any culture media, but some others need special conditions. So they require a special medium for their growth. Okay. Now, what are the conditions that should be contained in the culture medium? It should contain necessary nutrients, sufficient moisture, suitable pH. And the next thing, is this culture media, we are going to grow a certain type of microorganism that we want to study. Now just imagine, like, microorganisms, they are ubiquitous on Earth. They are found in everywhere, right? Can you hear me? But just imagine if your culture media, it contain like a contaminant microorganism or it contain like a bacteria or something. Huh? So are we going to study only about that certain type of microorganism that you want to study? No. We are going to study, then we if we take a sample of that culture media and if we observe it under the microscope, right, we are going to study about both, right, the microorganism that present in the culture media as well as the microorganism you cultured, right? Okay, so is that our our work, is it successful? No, it is not successful. So this is the thing, right? This is why we need something called sterilization. So before making this culture media, we have to sterilize all the glassware, right? That means the pipettes, petri dishes, right? Inoculating loops, everything that we use to make this culture media, as well as the culture media, right? Culture medium also we have to sterilize that solution. Okay, this is the point. This is the thing. This is why we need sterilization. This medium must initially be sterilized, which means it should not contain any living organism. Therefore, when preparing a culture medium, all glassware and liquid nutrients solution should be sterilized. Okay, can you understand? Can you understand what is culture media, about non-culturable and culturable microorganism, and why we need sterilization? If I summarize this part, this slide again, culture medium is, it is a nutrient material prepared for providing nutrition and encourage essential to the growth of microorganisms. So if you have to define this one, your definition should your definition should contain these two terms: providing nutrition and encourage essential, encourage, which is essential to the growth, right? Those are the points. 2 points should be included in your definition. Then the next thing is, some microorganisms are culturable, but some are non-culturable. And for the growth of some microorganism in the laboratory condition, we have to provide a special medium. And the next thing is that culture medium, it should contain necessary nutrients, sufficient moisture, and suitable pH, right? And for this culture medium, it should be initially sterilized, right? That means that it should not contain any living microorganisms, right? For that, we have to sterilize all the glassware and liquid nutrients that we are used to prepare this culture medium, right? Next one. Then, now there are two general media which are commonly used to grow bacteria and fungi. For the bacteria, we use something called nutrient agar, which is symbolized by capital Na. Nutrient agar medium. And for the fungi, we use potato dextrose agar, right? Now, if we take nutrient agar, the components of this nutrient agar culture medium is as follows, right? It contains peptone, right? Peptone, then meat extract, beef, right? Beef extract, sodium chloride, agar, and distilled water, right? What are the things, what are the constituent materials present in the nutrient agar? Peptone, beef extract or meat extract, sodium chloride, agar, and distilled water, right? We use nutrient agar medium to grow bacteria, right? That is for bacteria. And potato dextrose agar, right? That medium is used to grow fungi, and it is made up of potato, glucose, agar, and distilled water. Now if you compare these two, right? If you compare the nutrient agar and potato dextrose agar, right? You can see two constituents are common to both. What are those two constituents? What are the two constituents common to both mediums? Both media. Hmm? Huh. Aisha, Nisma? Agar and distilled water. We need agar and distilled water to produce both nutrient agar as well as potato dextrose agar. Right? Yes. Why? Now distilled water? It provides suitable moisture. But what is this agar? Now, if we take peptone, meat extract, right? They are nutrients. Here in the potato dextrose agar, potato and glucose, they are used as nutrients. But why? Well, why we have to use agar? What is the function of agar? What is the function of agar here? Why we have to use agar to produce this culture media? Now you can see it is common to both nutrient agar medium and potato dextrose agar. Right? Please respond, right? Aisha? Why? Yes. Agar, here agar it is used, we have to use it as a solidifying agent, right? Solidifying agent. Now these things are solutions, right? If we now meat extract, glucose solution, these are solution. Now we culture these microorganisms. Now we usually contain this, we store this culture media in a petri dish or test tube. Petri dish means something flat. නේද පීරිසියක් වගේ අර පොඩි ඒකේ පොඩි ගැට්ටක් තිබ්බට ඒක පීරිසියක් වගේනේ හරිද අන්න ඒවැ තමයි අපි මේ කල්චර් මීඩියා එක ස්ටෝර් කරන්නේ එතකොට අතක් වැදුණු ගමන් මොකද වෙන්නේ කෙලින්ම මේක ඉහිරෙන්න පුළුවන් it can split the culture media can split ඕකේ ඉන්න micro organism හැමතැනම ගිහිලා ඊට පස්සේ spread වෙන්න පුළුවන් right so to avoid that we have to solidify that right we have to solidify our culture medium එතකොට අර ගෙදර හදන ජෙලි එක වගේ මෙතන medium එක solidify කරන්නේ කවුද agar right agar is used as a solidified agent it is not a nutrient okay so mc2 point agar is not a nutrient ඒක nutrient එකක් නෙමෙයි ඒකෙන් කිසිම පෝෂණයක් ලැබෙන්නේ නැහැ it only act as a solidifying agent okay so at room temperature, right? It is a solid. It can solidify. So first what we have to do is, we have to heat it to temperature which is above 40 degrees Celsius. Then after that, we have to cool it at temperatures below 40 degrees Celsius, right? Agar, it solidifies at temperatures below 40 degrees Celsius. That means at room temperature, right? A culture media which contain agar is a solid medium. Okay. So that is about agar. The next thing is, now just imagine, right, okay, we culture microorganism in a culture media and then we have microorganism. Now we need to observe this microorganism under the, we don't have electron microscope in our labs. We have to observe microorganism in the compound microscope, okay? We have to observe the microorganism under compound microscope. But most of these microorganisms, they are colorless. Right? So can we observe like, that means we have to highlight them from the background to observe them more clearly. ඒගොල්ලන්ට පාටක් නැත්තම් අපිට පේනවද? background එකෙන් ඒගොල්ලන්ව වෙන් කරලා බලන්න පුළුවන්ද? බෑ. It's difficult. So we need to highlight them from their background. Right? For that, we have to use colors. We have to use dyes. And we have to color them. පාඩවෙන් සලවලින් චිත්‍ර පාඩ කරනවා වගේ ඩයි දාලා මේගොල්ලෝව පාඩ කරන්නයි යන්නේ. So this is called staining. මොකක්ද කියන්නේ staining. Staining means coloring the microorganism with a dye. Right? With a dye. Dyes. They are salts. හරි? Dye කියන්නේ salt එකක්. Now this part is not included in your syllabus. This is just for understanding, right? Okay? Salts, if we take salts, they are positive ions as well as negative ion. If the positive ion gives the color, it is called a basic dye. Now, for example, you know about copper sulfate. Copper sulfate is blue in color, right? It has a positive ion as well as a negative ion, right? Copper sulfate, it is not a dye, right? And I use copper sulfate just to explain. Now, what is the positive ion? Copper 2+. What is the negative ion? Sulfate. Now, here that blue color is resulted from which one? Due to that copper 2+ ion. Like that, dyes, they are salts. If the positive ion results the color, it is called a basic dye. And if the negative ion gives the color, it is called acidic dye. Now, in these salts, the color those pigments are containing something called chromophore. Chromophore කියන එක තමයි මේ colors තියෙන්නේ. Right? Then the next thing is there are two types of staining, positive staining and negative staining. Now, if we take a bacterial cell at pH 7, that means in neutral media, they are negatively charged. Bacterial cell එක neutral medium එකේ කියන්නේ pH 7 වලදී එයා negatively charged. හරිද? Okay? So, if we add a basic dye එකක් කියන්නේ මොකද්ද? Positive ion එක. Right? If we add a basic dye to color this bacterial cell, the bacterial cell is negatively charged and the dye is positively charged. Positive ion. So what will happen? Positive and negative ions, they will attract towards each other. So as a result, what will happen? The bacterial cell, it will stain, right? It will color. මොකද වෙන්නේ? බැක්ටීරියා සෙල් එක කලර් වෙනවා. අර බැක්ටීරියා සෙල් එක ගාව වටේට ගිහිල්ලා මේ බේසික් ඩයිස් ගිහිල්ලා කිහින් ඇට්‍රැක්ට් වෙනවා. So this is called positive staining. Okay? ඒකට අපි කියනවා positive staining කියලා. For positive staining we use crystal violet, methylene blue and safranin, right? These three examples are given in your resource book. And if the dye is a negative ion, and the bacterial cell is also negative now. What will happen? Negative charges, they repel each other. නේද? එහෙනම් bacterial cell එකෙන් මේ dyes මොකද වෙන්නේ? ඈතට යනවා. So as a result, the background will be stain, not the bacterial cell. තේරුණාද? මෙන්න මෙහෙමයි වෙන්නේ. මෙහෙම ඉන්නවා bacterial cells. මේගොල්ල මොකද? negatively charged. මේ අපේ dye එක එනවා. ඒගොල්ල මොනවද? Sorry. They also negatively charged. So they will repel from the bacterial cell. As a result, what will happen? This background, it will be colored or stained. So this is called staining, like negative staining. Here the background is staining, not the bacteria. Bacterial cells, they remain colorless. Okay? Can you understand? So eosine and nigrosine are two examples for dyes, right? Acidic dyes. Okay? Can you understand? Any doubts? Any doubts? No. Please reply. No. Nisma, what about you? Any doubts? Okay. Then, so bacteria cells like microorganisms, they appear almost colorless when we weave through the compound microscope. So we have to prepare them for observation. And so the one way of doing that is we have to stain them. Staining means we have to color the microorganism with a dye, right? Anyway, before staining, right, we have to fix this microorganism or attach the microorganism to a microscopic slide, right? Now, you have cultured a microorganism in a culture media. The culture media is present in a petri dish. From the petri dish, you have to transfer like a small amount of microorganism onto a slide, right? Onto a glass slide. Natham petri dish, you can't keep it on the compound microscope and observe it. You have to make a slide, right? You have to transfer the specimen onto the glass slide, right? Here, first we have to, before staining, now we are staining the specimen, right? Not the culture media. Hari culture media එකට ගිහිලා dye එක දාන්නේ නැහැ. අර හදාගන්න glass slide එකේ ඒ තියෙන specimen එක තමයි color කරන්නේ. So before adding dye, we have to fix the specimen, right? We have to fix the microorganism to the slide. හරිද? Right. We can discuss about those things later, right? In the practical parts. So simple stain, right? Simple stain, it is an aqueous or alcohol solution of a single basic dye, right? The primary purpose of this simple stain is to highlight the entire microorganism, right? We have to stain because we need to highlight the entire microorganism to study its structure, I mean the cellular shape and the cell arrangements and cellular shape and the cell arrangements. And so in the laboratory, we use these dyes, crystal violet, methylene blue and saffron, right? We'll move to the note again. Right, okay, look at this one. Because most microorganisms appear almost colorless when viewed through a standard light microscope, we have to prepare them for observation. One of the ways this can be done is by staining, means coloring the microorganism with the dye. However, before microorganisms stain, they must be fixed to the microscopic slide. A simple stain is an aqueous or alcohol solution of a single basic dye. The primary purpose of a simple stain is to highlight the entire microorganism so that cellular shape, cell arrangement, and basic structures are visible. Some of the simple stains commonly used in the laboratory are methylene blue, crystal violet, and saffron, right? Next one. So this is a culture media stored in a petri dish. Right. Now we are going to start practical part. Sterilization of water, culture media, glassware, heat labile substances and inoculating needles, right? This is practical, I think 35 in your practical guide. Yeah, sterilization by dry heat using direct flame, right? Dry heat using direct flame. Can you remember when we discuss in that theory note, right? How we can do this? We have to hold the inoculating loops or needles to the Bunsen flame until they reach red hot, right? When we hold these inoculating loops and needles to the Bunsen flame until the instrument, like if we take the inoculating loop, if we hold it until it reaches red hot, what will happen? The microorganism, they burn to ash, right? They burn to ash. Now here the practical part. For inoculating needles, loops and materials which not be damaged by heat, hold in flame of Bunsen burner until red hot. Right? Again, inoculating loops, needles, right? ඒවා මොකද කරන්නේ? කෙලින්ම Bunsen burner එකට අල්ලන් ඉන්නවා. Red hot වෙනකම්. But scalpels, metal spatulas, glass rods, right? We are not going to hold them until they reach red hot. to the Bunsen burner, right? Bunsen flame එකට ඒගොල්ලෝ මේ red hot වෙනකන් අල්ලන් ඉන්න යන්නේ නැහැ. Here what we have to do is we have to dip these in things in the methylated spirits or ethyl alcohol. And we have to drip off the excess and we have to flame the instrument in the Bunsen flame, right? We are not holding the instrument to the flame of the Bunsen burner until they reach red hot, right? Here there is a difference. Scalpels, metal spatula, glass rod, මොකද කරන්නේ? Methylated spirits වල හරි ethyl alcohol වල හරි පොගෝලා වැඩිපුර තියෙන ටික drip off කරලා නිකම් මේ flame එකට ටිකක් අල්ලන් ඉඳලා අරගන්නවා. හරිද? Next thing, sterilization by dry heat in the hot air oven, right? For sterilization of dry glassware such as petri dishes, flasks, and pipettes, prepare glassware for sterilization as follows. Sterilization එකට instruments ටික කොහොමද අපි හදන්නේ කියලා මෙතන අපි ඉගෙන ගන්න. Prepare කරන්නේ කොහොමද? Right? What we are using hot air oven, we have to keep this glassware inside the hot air oven, but before that we have to prepare them. නිකම්ම ගිහිල්ලා ඇතුලෙන් තියන්නේ නැහැ. ඒකට අපි ඒක ඒ කියන්නේ ඒවා prepare කරන විදිහක් තියෙනවා. Now dry heat this hot air sterilization කියන එක මොකක් යටතේද ආවේ අපේ? Hot air sterilization we learn under dry heat sterilization, right? So here first we have to wash the glassware and we have to clean and wipe dry thoroughly, right? හොදලා ඉස්සෙල්ලා මේක හොඳට පිරිසිදු කරලා පිහිදාන්න ඕනේ. තෙත නැති වෙනකම් පිහිදාන්න ඕනේ. තෙත මාත්තු කරන්න ඕනේ. Now if they ask us to explain the sterilization method, right? Explain about sterilization methods, you don't have to include these practical parts, right? But like Karanigang, if they ask you to explain about hot air sterilization, I think it's better to include these procedures also, right? Nikang are overall local as we discussed in the last session, in the session 3, then no need. හරිද? එතනම points ගොඩක් තියෙනවානේ. හැබැයි වෙනම දීලා තිබ්බොත් I think it's better to explain these procedures also. Okay, Aisha? Right. So using hot air sterilization normally, right? We can sterilize glasswares like petri dishes, flasks, beakers, bottles, and glass pipettes, right? We are not going to sterilize solutions containing vaccines, protein solutions using this method, right? Yes. So first we have to wash the glassware, we have to clean and we have to wipe dry. Then we have to wrap the glassware in aluminium foil or in a paper or store in a suitable container. කියන්නේ aluminium foil තියෙන්නේ. දන්නවනේ? aluminium foil එක හරි පත්තර ගොල වලින් හරි එහෙම නැත්නම් canister එක හරි මේවා store කරන්න ඕනේ. Another glassware sticker. Now, conical flask, if we take conical flask, they have a mouth like part. So plug the mouth with clean cotton wool and we have to cover the plug with aluminium foil. Eke cutter, මොකද කරන්නේ? Cotton wool ඔබලා වහනවා. වහලා ඒ plug එක තියෙනනේ ඒක aluminium foil එකකින් ඔතනවා. Then pipettes, right? Pipettes we have to plug them out with cotton wool and we have to burn the cotton fibers sticking out using a Bunsen flame, right? අර එළියට දික් වෙලා තියෙන cotton wool කෙදි ටික තියෙන්නේ ඒ ටික Bunsen flame එකක් අල්ලලා පුච්චලා දානවා. හරිද? ඊට පස්සේ we have to wrap the pipettes individually in aluminium foil or paper, or we can store them in a canister. Something looks like a thermoflask. මේති canister එකට වෙනම දාන්න පුළුවන්. Right? So here what are the conditions we use? What is the temperature? 170 degrees Celsius, and we have to maintain for 2 hours, right? Based depending on the amount of glassware we have to sterilize here. And the And we have to keep the oven door tightly closed. Right. Well, for sterilization of dry glassware such as petri dishes, flask and pipette, prepare glassware for sterilization as follows. ඉස්සෙල්ලා මොකද කරන්නේ? We have to wash the glassware clean and we have to wipe it. Then we have to wrap the glasswares in aluminium foil or wrapping paper, store in a canister. Now, if we come to conical flasks, we have to plug them out with cotton wool and we have to cover the plug with aluminium foil. Then for pipettes, we have to plug the mouth with cotton wool, and the cotton fibers which sticking out of the mouth, we have to burn them using a Bunsen flame. And we have to wrap the pipette separately using aluminium foil or paper. We have to store them in a canister, right? And after storing all these prepared glasswares in an oven, we have to maintain 170 degrees Celsius temperature for 2 hours, depending on the amount of glasswares in the oven. And the door of the oven should be closed tightly. Okay, Harida? Next one. Sterilization in an autoclave, right? Autoclave, wet heat. Moist heat in moist heat sterilization, we learned about this one, right? In the moist heat sterilization, what is the instrument we have to use? We have to use an autoclave, right? Normally, this moist heat sterilization is used to sterilize culture media, right? Solutions, syringes, needles, and healthcare instruments, right? And like various instruments that can withstand at high temperatures and pressures. What are the conditions we are using here? What is the temperature? 121 degrees Celsius. And the pressure is 15 psi, right? Pounds per square inches. Or 1 atm, right? For how many minutes? For 15 minutes, 15 to 20 minutes, right? First, now look at this procedure. Place the prepared liquid culture media or water in test tube, flask or bottles appropriate. Right? එතකොට මේකටත් කමුලින්ම ඉස්සෙල්ලාම test tube, flask ඒ ටික මොකද කරන්න ඕනේ? First we have to sterilize them. ඒ ටික sterilize කරන් ඉන්නවා අර කලින් විදිහට. Then after that we have to place our prepared liquid culture media or water in that test tube, flask or bottles. Right? Now, just imagine like you stored a culture media in a flask. Then you have to plug the mouth of the flask with cotton wool, and we have to cover it with aluminium foil or paper. Right? If we have like bottles with screw caps, then හිතන්න මෙගා බෝතලේ screw cap එකක් කියන්නේ මොකද? අර බෝතල් මූඩිය කරකවලා අරින එකන්නේ. screw cap එකක් තියෙනවා නම්. එහෙම බෝතල් තියෙනවා. We have to lose the screw cap slightly, because what will happen here when we provide these conditions, right? Here wet heat, the air present inside these bottles, it will expand. මේවා ඇතුලේ තියෙන වාතය මොකද වෙන්නේ? expand වෙනවා. expand වෙලා ගලන්න යන්න ඉඩ නැතුනොත් මොකද වෙන්නේ? බෝතලේ පුපුරනවා. So to avoid that, we have to loose the screw cap slightly, and we have to place the containers in the autoclave. Close the lid of the autoclave tightly and open the valve. Right? මුලින්ම මොකද කරන්නේ? pressure cooker එකේ වගේමයි. ඉස්සෙල්ලාම we have to place all the things in the autoclave, and we have to close the lid of the autoclave tightly. And there is a valve. සිංහලෙන් කියනවා කපාටය කියලා. valve එක අරිනවා. ඇරලා when we provide this condition, when we heat it, what will happen? You can see water vapor, it released from the valve. When water vapor start to release out of the valve, we have to close the valve. valve එක වහලා දාන්න. හරි? water vapor එළියට එන්න, ජල වාෂ්ප එළියට එන්න ගත්ත ගම valve එක වහලා දානවා. හරිද? valve එක වහලා ඊට පස්සේ ඔය කියන time එකේ තියලා ඊට පස්සේ ආයෙ මොකද කරන්නේ? එකපාරක් ගිහින් lid එක අරින්නේ නැහැ. pressure cooker එකත් උනත් එකපාර අරින්නේ නැහැ නේ. මොකද the inside that the pressure is high. Right? It is filled with water vapor. වතුර වාෂ්ප තියෙනවා. එකපාර ඇරියොත් මොකද වෙන්නේ? අර ටික ඔක්කොම ඇඟට එන්න පුළුවන්. එතකොට ආපහු වෑල්ව් එක ඇරලා අර තියෙන නෝම් ඒ කියන්නේ මේ ප්‍රෙෂර්ස් දෙක ඉක්වල් වෙන්න දෙනවා. එළිය ප්‍රෙෂර් එකට ඇතුලේ ප්‍රෙෂර් එක ඉක්වල් වෙන්න දෙනවා. ඔක්කොම water vapor වලට යන්න දීලා after that you have to open the lid and you have to take those whatever the glasswares you sterilize out. Right? Sterilization in an autoclave. Wet heat. For sterilization of water, culture media prepare the glassware for autoclaving according to the procedure outlined above. Place the prepared liquid culture media or water in test tubes, flask or bottles as appropriate. Plug the containers with cotton wool and cover with aluminium foil or paper. If bottles with screw caps are used, now the thing is you have to study these things because in the structured essay, they can ask you to explain these things, right? Explain the procedure that you have, procedure that you have to follow to sterilize whatever the culture media in an autoclave. Autoclave එක දාලා culture media එක sterilize කරන procedure එක explain කරන්න කියනවා. So you should study these things. තේරුණාද? Okay, Aisha? And for the MCQs also they can give. හරි? Close the lid of the autoclave tightly and open the valve. Set the pressure at 15 psi and heat. Close the valve when water vapor is released. Autoclave for 15 to 20 minutes at 121 degrees Celsius. Right? I will include this kind of question in our special paper series. Right? Okay. Next one, sterilization by filtration using membrane filter apparatus. Right? Right. You know, for solutions like temperature sensitive solutions, like solutions which contain vaccines, enzymes, proteins, right? We can't use the above two methods to sterilize. We can't use any dry heat sterilization or moist heat sterilization, right? Because what will happen if we hold them to the flame, right? Or if we provide high temperatures, all those vaccines, proteins, they will denatured, right? Yes. For that kind of things, that kind of heat labile substances, we have to use this method. Sterilization by using filter method or membrane filtration. Can you remember the range of, the size range of pores of these membrane filters? Pores of membrane filters, the range, what is the range? 0.01 micrometers to 0.45 micrometers size. So these membrane filters used to sterilize solutions contain enzymes, vitamins, antibiotics, vaccines, and some culture media, right? And here, the material to be sterilized is passed through the filter by using a vacuum, right? Vacuum එක තමයි මේක sterilize වෙන්නේ. Pass කරන්නේ vacuum එක. ඒක explain කරා in the session 3. Then this filter, it remains the microorganism and liquid pass through the filter. Using this method, we can in this filter, it retains all the microorganisms, including these viruses as well as large protein molecules. Yes. Now look at this one. Sterilize the components of the membrane filter apparatus separately. Right? Take it, use kind of funnels, flask. We have to sterilize those components separately. After that, filter the liquid using membrane filters. Collect the filtrate into the sterilized bottle or test tubes. Right. Now, preparation of a microbiological culture medium. Nutrient agar, potato dextrose agar, inoculation with a sample of toddy and yogurt and observation of microbial colonies, right? This is practical number 36. Yes, now we are going to make a culture media. We are going to prepare two culture media. One is nutrient agar and the other one is potato dextrose agar. Nutrient agar is used to grow which kind of microorganism? To which type of microorganism? Viruses, fungi, or carbon? Bacteria, very good. Right? We use nutrient agar to grow or to culture bacteria. හරිද? මේ බාන් දීලත් මේකේ තියෙන දේවල් peptone කොච්චරද 10 grams. Beef extract 10 grams. Sodium chloride 5 grams. Agar 15 grams. Distilled water 1000 milliliters. Right? This is given in your practical manual. And potato dextrose agar used to culture which kind of microorganism, which type? What is the type of microorganism growing this potato dextrose agar culture media? Nisma? Fungi. Very good. Fungi. Potato dextrose agar for fungi. Potato, glucose, agar and distilled water. Here potato and glucose they use as nutrients. Agar as the solidifying agent and distilled water to provide sufficient moisture. Right. First we have let the students prepare nutrient agar and PDA from the given material. හරිද? solution එක now we prepared the solution to culture media. Right? Sterilize the solution by autoclaving at 121 degrees Celsius for 15 minutes at 1 atm pressure. We have to sterilize those two solutions. Assist the students to prepare nutrient agar and PDA plates as given below. Then solutions දෙක හැදුවා, මේ දෙක sterilize කරා. ඊළඟට මොකද කරන්න ඕනේ? අර දැන් ඒව microorganisms ලා මොකෙක්වත් නැහැනේ. හරිද? හිතන්න අපිට ඕනේ කියලා මොකෙක් හරි fungi කෙනෙක් observe කරන්න කියලා. I think sample එකක් ගන්නවා. හිතන්න toddy sample එකක් ගත්තා. ඒක ඉස්ලා ඉන්නවනේ. Right? අරගෙන මොකද කරන්නේ? Now here first you have to pour 50 milliliter of sterilized nutrient agar and PDA into sterilized petri dishes using aseptic techniques. Right? Set aside to solidify. Solutions දෙක එතන දැන් මෙතන අපි temperature එක මොකද කරන්නේ 121 degrees Celsius temperature එකක් දෙන්නේ. So definitely this solution it is a liquid thing, right? Not a solidified. It's not a solid medium because agar you know it solidifies at temperatures below 40 degrees Celsius. Now here the temperature is 121 degrees Celsius, right? 3 times greater than that. එහෙනම් මෙතන අනිවාර්යයෙන් මේක solution එකක් විදිහට තියෙනවා. හරි. sterilize කරපු ඒ solution එක අරන් ඒක අපේ petri dishes වලට දානවා. දාලා ඔන්න පැත්තකින් තියනවා, right? When that temperature drops to room temperature, what will happen? When it drops below 40 degrees Celsius, that agar containing this culture medium, they start to solidify. Okay? තේරුණාද? Can you understand? එතන වෙන දේ? ඊට පස්සේ right? දැන් ඔන්න අපි ගාව තියෙනවා culture media එක. ඒ කියන්නේ petri dishes වලට දාගත්ත culture media එක. දැන් හිතන්න අපි ගත්තොත් nutrient agar එකේ අපි bacteria නේ grow කරන්නේ. Right. අපි ගන්නවා yogurt sample එකක්. yogurt වල microorganisms ලා නේ bacteria ඉන්නවනේ. හරිද? ඊට පස්සේ ඉස්සෙල්ලාම first we have to label the bottom of this agar plate using a marker pen. හරිද? petri dish එකේ bottom එක. ඒ කියන්නේ අර යට තියෙන මේ අපේ culture medium එක දාපු කෑල්ල දැන් ඒකේ කොටස් දෙකක් තියෙනවානේ. පියනක් lid එකක් තියෙනවා. වෙන මේ plate එක තියෙනවා. Here we have to label the bottom of the agar plate, not the lid. Because why? The lids they can change. The lids මාරුනොත් මොකද වෙන්නේ? හිතන්න lid දෙක මාරු කරොත් nutrient agar culture medium එකේ potato dextrose එකකින් මොකෝ කාලා බලන්නද? නේද? එහෙනම් bottom එක petri dish එකේ යට පතුල තමයි අපි label කරන්නේ. හරිද? ඔන්න label කරනවා bacteria හරි මොකක් හරි label කරනවා yogurt sample හරි ගාලා label කරනවා. ඊට පස්සේ we have to take an inoculating loop, right? Loop, and we have to sterilize it. How we can sterilize it? What is the method we have to use here? Can we do it by moist heat sterilization or filtration or pasteurization? What is the method used to sterilize inoculating loops? Here, Nick turn on your mic and tell me the answer. Aisha, Nisma? Yes, by direct framing. You can hold the inoculating loop to the flame of Bunsen burner until it reach red hot, right? After that, you can obtain a loop full of that yogurt sample. Loop එක ඉස්සෙල්ලම sterilize කරලා ඒකේ සම්පූර්ණ يعني loop එක full වෙන්නම sample එකක් අරගන්නවා. right? හිතන්න yogurt ගත්ත කියලා. හරිද? Now you have the petri dish and you have a loop full of sample of yogurt. What you have to do? You have to transfer them onto the petri dish, right? Place a loop full of sample on the agar plate near the edge of the dish and streak on the agar surface in a zigzag pattern. It's very important. ඔහේ නිකන් පිස්සෙක් වගේ යදින්නේ නැහැ මේකේ. හරිද? මේකට petri dish එක නම් මේක edge එක තියෙනවානේ. edge එක ගාවින් මෙහෙම loop එක තියලා zigzag pattern එකට තමයි ඇඳගෙන යන්නේ. එතකොට දැන් මේ මොනවද මේ කල්චර් මීඩියම් එක ගෑවෙන්නේ? යෝගට් නේ අපි ගත්තේ. යෝගට් ඔන්න තීරුවක් කොහොම ගෑවිලා යනවා. ඊට පස්සේ මේක ඉන්කියුබේට් වෙන්න දුන්නාම මොකද වෙන්නේ? මේකේ මයික්‍රෝ ඔර්ගනිසම් ග්‍රෝ වෙනවා. නේද? ඊට පස්සේ එතනින් චුට්ටක් අරගෙන අපි කරන්නේ මොකද්ද? ස්ලයිඩ් එකකට දාලා ස්ටේන් එයාව හීට් ෆික්ස් කරන් ස්ටේන් කරලා කම්පවුන්ඩ් මයික්‍රොස්කෝප් එකේ දාලා ඔබ්සර්ව් කරනවා. ඒකනේ කරන්නේ. But here, what is the process you have now when you are transferring the sample from the source to the culture media, you have to use this step. You have to use this method. You have to keep the loop full of sample near the edge of the petri dish and you have to streak on the agar surface in a zigzag pattern. After that, we have to incubate for 24 to 48 hours, right? At room temperature. Then after that time period, you can observe the colonies on incubated plates, right? Normally, then is called in our school when we are doing this practical, we did this one on a Friday, right? Then again in the next week, right? On Monday, we observe this microorganism under the microscope. weekend එකේ දවස් දෙකක් තිබ්බනේ ඒගොල්ලන් ඉන්කියුබේට් වෙන්න. right? හරිද? හරි නේද? ඒක තේරුණාද එතන? Okay. So this is called inoculation of plagues. Right. Nisma, what about you? Any doubts? Right, Mithi. The streak plate isolation method. Now first, hold the inoculating loop against the flame of the Bunsen flame until it turned red hot. Right? Then take a loopful sample of yogurt. Yogurt gatta කියලා. Then place the loopful sample near the edge of the petri dish and streak zigzag patterns. right? එතකොට දැන් එකපාර නිකන් අර මං ඇඳා වගේ ඇඳොත් මොකද වෙන්නේ? ලොකු කොලනි එකක් හැදෙනවා. එතන ලොකු මයික්‍රෝ ඔර්ගනිසම්ස්ලා ප්‍රමාණයක් හැදෙනවා. Then it's difficult for us to separate like a colony from that. right? ඒකෙන් චුට්ටක් ඒ කියන්නේ separate කරගන්න බෑ. ගුලිය වගේ ඉන්නවා. එතකොට එහෙම එකක් මයික්‍රොස්කෝප් එකේ දාලා බලුවම හොඳට observe කරන්න බෑ. වෙන වෙනම බලන්න අමාරුයි. එතකොට අපි කරන්නේ මොකද්ද? ඉස්සෙල්ලාම ගන්නවා. ඉස්සෙල්ලාම ඔන්න ඔය විදිහට zigzag pattern එකෙන් අඳිනවා. ඊට පස්සේ ඔය loop එක තියෙනවනේ. හරිද? ඒ කියන්නේ අපි දැන් sample එක අරගන්න ඒ කියන්නේ දැන් culture medium එකේ incubate උනන් පස්සේ no sorry ඉස්සෙල්ලාම ඔන්න sample එක තියෙනවනේ. ඒකේ අර විදිහට loop එකෙන් අරගෙන අඳිනවා. inoculating loop එකෙන් අරන් මේ විදිහේ zigzag pattern එකකට අඳිනවා. හරිද? මෙන්න මේ වගේ. මේ බලන්න. මෙතන. like this one. right? Then we have to hold the inoculating loop to the Bunsen flame, right? Bunsen flame. And again, you have to streak this zigzag pattern on the culture medium, in the agar medium, right? එතකොට දැන් ආපහු අර source එකේ ඔබලා අරගන්නේ නැහැ. ඔය විදිහට මෙන්න මේ මෙතන මේ තින් පැත්තෙන් එහෙම අඳිනවා. තුන්වෙනි වතාවටත් එහෙම කරනවා. ඒ කියන්නේ ආපහු බන්සන් ෆ්ලේම් එකට අල්ලලා ස්ටෙරිලයිස් කරලා ලූප් එක ආපහු අදිනවා. හතරවෙනි වතාවටත් ඒ විදිහමටම කරනවා. එතකොට මොකද වෙන්නේ? නිකන් ඔයගොල්ලෝ මාක පෙන් එකකින් ලියාගෙන ගියොත් බෝඩ් එකේ පටන් ගන්න තැනදී කොහමද? හොඳ තික් එකට තදට ලිය වෙලා තියෙනවා. අන්තිමටම ලියද්දී කොහමද? ඇහැට පේන්නේ නැති ගන්න ලාවට නේද ලිය වෙන්නේ? නේද? Have you experienced that kind of thing? මාක පෙන් එකකින් ලියද්දී මුලින් හොඳට ඉන් තියනවා. හොඳට පේනවා. අන්තිම යන්නේ ස්කෝලේ එහෙම දැකලා නැති. You have to imagine what is there. What is on the board. Have you experienced that kind of thing? Aisha? Right, yes. So the same thing happened here. මුලින්ම ගාද්දි මොකද වෙන්නේ? මයික්‍රෝ ඔර්ගනිසම්ස්ලා හොඳට අර යෝගට් සැම්පල් එක හොඳට ගෑවෙනවා. අර විදිහට දෙතුන් පාරක් ස්ටෙරිලයිස් කරලා ගාද්දි මොකද වෙන්නේ? අන්තිමට යද්දි චූට්ටයි ගෑවෙන්නේ. ඒකට චූට්ටක් ඉන්න තැන ඉන්කියුබේට් කරලා ආවෙන් පස්සේ කොලනි එක එච්චර ලොකුවට තියනවද? නෑ. එච්චර ලොකු කොලනි එකක් එතන නෑ. Right? Then it is easy for us to separate. මේ තියෙන්නේ එහෙම එකක්. Now this is the first quadrant, second one, third one. Now look at the fourth one. Look at the fourth one. Make a. Here, these sphericals are the bacterial colonies. Right? මෙන්න මේ වගේ තැනකින් ගන්නවට වඩා මෙතනින් ගත්තාම then it is easy for us to observe. Right? Under the microscope. Right. Staining microorganism found in toddy and yogurt using a simple stain. Methylene blue. Here we use methylene blue as the dye. Is this a positive staining or negative staining? This is a basic dye. So this is a positive stain. That means the dye is positively charged and now if we take bacterial cell, they are negatively charged. Preparation of smear. Clean the slide with a cleanser, rinse and air dry. Handle the clean slide by the edges, preferably using a pair of forceps. You have to take a glass slide and clean it and let it air dry. Then don't handle the clean slide by your hand. You have to hold it by the edges or you have to use forceps. Then using a marker pen, you can label the slide, right? You can label it. Then for the yogurt sample, place 1 or 2 loops full of distilled water on the center of one slide using the sterilized inoculating loop. Now you have the clean glass slide and using a sterilized inoculating loop, keep 1 or 2 loops full of distilled water at the center of the glass slide. Okay, then you have to hold the inoculating loop to the Bunsen flame until it's reached red hot. Then allow it to cool and then scrape a small amount of sample using the cooled loop. That means from that culture media. Then emulsify the scraping. Emulsify means you are dissolving or you have to transfer that sample onto that water drop which is on the middle of the glass slide, and you have to spread the suspension in a shape of a circle. Here the smear it should be very thin. So this is called emulsifying, right? Okay, that is for the yogurt sample. For the toddy it contains yeast, right? Here we don't have to use water. We don't have to keep a water drop at the center of the glass slide because now if we take toddy, it is already it is a liquid thing, right? So the microorganisms, they are already suspended in water. So we have to sterilize the inoculating loop and we have to scrape small amount of we have to take some toddy and we can transfer it to the glass slide and we can observe it under the microscope. So before observing under the microscope, we have to stain it. So this is common to both yogurt sample and toddy sample. First, we have to heat fix the smear. That means we have to attach the smear to the slide, right? Here what we have to do is, on a glass slide, we have to hold it using forceps or something, and we have to pass this through the Bunsen flame 2 or 3 times. මොකද කරන්නේ? forceps එකකින් අර slide එක අල්ලලා කෙලින්ම flame එකට අල්ලන් ඉන්නේ දෙතුන් පාරක් එහෙට මෙහෙට flame එක හරහා ගෙනියනවා. තේරුණාද? After that, right, we have to put 2 or 3 drops of methylene blue to get heat fixed smear and allow 30 to 60 seconds for the stain to act, right? Flood the prepared heat fixed smear with 2 or 3 drops of methylene blue and allow 30 to 60 seconds for the stain to act. After that, we have to wash the excess stain, right, with tap water. And we have to blot the smear with blotting paper and we have to let it dry. Tissue paper use කරලා we have to blot it. After that, we can examine the stained smear under the microscope, right? So this is the method how you can observe microorganism under the compound microscope, right? So we discuss about methods of sterilization, why we need sterilization, how to sterilize glasswares, how to prepare the culture media, and how to sterilize culture medium, and inoculation of plates. That means how to transfer the sample to the culture media. Then, right? Then heat fixing and staining of the smear. Okay, any doubts? That part is over. Any doubts? ප්‍රශ්න මොකුත් තියෙනවද? නිස්මා? Aisha? Right. Okay. Now, let's start next part, microorganisms and diseases. Okay, session 4. No. Microorganisms and diseases. Right. Normally, right, humans, they are free of microorganisms at birth. Right? Generally. Now, the word generally is important, but not always. That means generally. ඒ කියන්නේ එහෙම contaminate microorganisms ලා ඉන්නත් පුළුවන්. Right? You know the baby when it is inside the mom's womb, it is connected with the mom, with the mother, with the umbilical cord. So there are some microorganisms associated with the umbilical cord, right? However, during birth, the newborn first get in contact with microorganisms present on the vagina of the mother, right? There are lactobacillus, right? Lactobacilli bacteria They colonize in the vagina of like females. So during the birth, during a normal like natural birth, right? The newborn, they can get contaminated with the microorganism that present on the vagina of mother. That is lactobacilli here. The lactobacilli, it should be italicized, right? Typing mistake. So lactobacilli colonize the intestine of the newborn. After birth, many other microbial populations begin to establish inside or on the surface of the body. Inside means inside our digestive system. That means inside the large intestine, right? Like that. And on the surface of the body, right? On the skin surface. Okay. So these are called normal microbiota of human body. Anyway, right? However, the internal tissues, right? Internal tissues of the healthy human body are free of microorganisms. මොකක්ද ඒ කියන විකාර කතාව බලන්න. ඇතුළේ microorganisms ලා ඉන්න පුළුවන්. හරිද? මේකට කියනවා normal microbiota කියලා. ඊට පස්සේ කියනවා internal tissues වල microorganisms නෑලු. Right? Give me a second. Right. Internal tissues. Here, internal tissues means the brain, the nervous tissues, and the lungs, right? The microorganisms, they can present in our nasal cavity, right? But not in the lungs. If they present in the lungs, then that the man is not healthy, right? Okay. So here, internal tissues means the lungs, right? The brain tissues, brain cells. No, that means the neurons, like those things. Okay. Can you understand? Now, let's read the paragraph. Generally, humans are free of microorganisms at birth. However, during birth, the newborn first get in contact with the microorganism present on the vagina of mother. Usually, these are lactobacilli. Lactobacilli colonize the intestine of the newborn. After birth, many other microbial populations begin to establish inside or on the surface of the body. These are called normal microbiota of human body. However, internal tissues of healthy human body are free of microorganisms. Okay? A part of the microorganism colonize on the skin and majority enter the body and colonize on the inner surfaces such as mucous membranes of nose, throat, upper respiratory tract, intestinal tract, and genitourinary tract. Right? Yes. Next one. A normal healthy body, it contains large number of microorganisms, right? And now here they have given a comparison. If it is estimated that the human body, it is like if the total number of body cells is 1 into 10 to the power 13, the number of microbial cells associated with the body is 1 into 10 to the power 14. Right now you can see the number of microbial cells is 10 times greater than the number of human cells. අපේ ඇඟේ තියෙන සෛල වලට වඩා 10 ගුණයක් microbial cells අපේ ඇඟේ ඉන්නවා. තේරුණාද? Right? හරිද? But these microorganisms, majority of these microorganisms, they are either harmless or beneficial. Right? Beneficial. Now, for example, if we take Escherichia coli, E. coli, right? The minimal colonization of this E. coli, right, in the large intestine It prevents colonization of pathogenic bacteria like Salmonella typhi. Again, here Salmonella typhi should be italicized, right? Salmonella typhi. Okay, and E. coli in the large intestine, it can synthesize vitamin K and some vitamin Bs, which are absorbed into the bloodstream and used by body cells. Okay, here now the word minimal colonization is important. Minimal colonization of Escherichia coli at large intestine prevent colonization of pathogenic bacteria. Okay, so is that E. coli is beneficial or harmful? Is it beneficial for us? Yes, now here it is beneficial. But the thing is, if this E. coli, right, it enters into our urinary bladder or lungs, it can cause diseases. When it remains in the large intestine, it is beneficial, not harmful. හැබැයි බැරි වෙලා හරි urinary bladder එකට lungs වලට එහෙම ගියොත් මොකද? එයා යකා නටනවා. නේද? හරිද? So they are called opportunistic pathogens. Okay? Can you understand? Opportunistic pathogen. Right? හරිද? Oh, please respond. Aisha, Nisma, okay? Can you understand who are pathogenic, opportunistic pathogens? Opportunistic pathogens means, yeah. Opportunistic pathogen means when they can change their interaction with the human body based on the conditions and they can cause infection. The example is E. coli. Once they remains in the large intestine, they are beneficial for us. But once they get into the urinary bladder, they can cause urinary tract infections. And if they enter into the lungs, they can cause pulmonary infection. Right. Then, probiotics. Probiotics. අර ටීම් එක දිහා බලන් ඉඳලා probiotics ගැන දන්නේ නැති. yogurt එකක් එහෙම මාකට් කරන්නේ. අපේ yogurt එකේ මේ probiotics තියෙනවා, මේවා තියෙනවා, අරවා තියෙනවා එහෙම කියලානේ කියන්නේ. Probiotics are live microbial cultures. So yogurt, it contains probiotics, right? So they are live microbial cultures. Ingestion. So here, what is the use of these probiotics? They prevent colonization of this Salmonella enterica, right? And this normally, for example, the ingestion of this lactic acid bacteria. Probiotics are lactic acid bacteria. They can alleviate diarrhea and prevent colonization by Salmonella enterica during antibiotic therapy. Now, if we take yogurt products in our country, like there are different products, Noodle kiyai, Palavatta kiyai, now, if we compare the taste and the color, they are different, not the same. Right? The difference in color or taste is due to the probiotics they used. Right? ඒගොල්ලෝ use කරන probiotics ඉන්න තමයි මේ difference එක තියෙන්නේ. Noodle පාවිච්චි කරන probiotic එක නෙමෙයි පැලවත්ත ගාව තියෙන්නේ. පැලවත්ත Highland එකේ වැඩි. එතකොට ඒකින්ද තමයි color එක ඊට පස්සේ taste එක වෙනස් වෙන්නේ. හරිද? Right. Now, let's go through the resource book note. A normal healthy body contains a large number of microorganisms. It has been estimated that the human body consists of 1 into 10 to the power 14 microbial cells for 1 into 10 to the power 13 total body cells, which means that 10 times more microbial cells than human cells. Majority of these organisms are generally harmless or even beneficial. For example, minimal colonization of Escherichia coli at large intestine prevent colonization of pathogenic bacteria such as Salmonella typhi. E. coli in large intestine synthesizes vitamin K and some of the B vitamins that are absorbed into the bloodstream and used by body cells. Recent interest in the importance of bacteria to human health has led to study of probiotics. Probiotics are live microbial cultures. Example, yogurt exert a beneficial effect. Several studies have shown that ingestion of certain lactic acid bacteria can alleviate diarrhea and prevent colonization by Salmonella enterica during antibiotic therapy. Although majority of human microbiota are harmless, some of them may change their interactions with human body under certain conditions and cause infections. Those microorganisms are called opportunistic pathogens. For example, E. coli is generally harmless as long as it remains in the large intestine. However, they may cause diseases. If enter other body parts, for example, if they enter into the urinary bladder, they cause urinary tract infections. And if they enter into the lungs, they cause pulmonary infections. Okay. Methane probiotics, lactobacillus, lactococcus, propionobacterium, right? Yes. Right. Next one, you have to study these four terms. You should know the definition of pathogen, host, pathogenicity, and parasite. Pathogen. Pathogen is an organism or entity that is capable of causing diseases. ලෙඩේ හදන්න පුළුවන් එක නට අපි කියනවා pathogen කියලා. Host, organism within which infected pathogens live or in and multiply. Right? Pathogenicity, the ability of a pathogen to cause disease in the host by overcoming the defense of the host. An organism or entity living on or in another living organism and obtain nutrients and other resources for the host. So pathogen is an organism or entity that is capable of causing diseases. Host is the organism within which infected pathogens live on or in and multiply. Pathogenicity is the ability of a pathogen to cause diseases in the host by overcoming the defense of a host. Parasite is an organism or entity living on or in another living organism and obtain nutrients and other resources from the host. Right? Okay. Right. Then, characteristics of pathogenic microorganism. Pathogenic microorganism කියන්නේ එයා තමයි එයාට පුළුවන් ලෙඩේ හදන්න. එහෙමනම් දැන් එයා ලෙඩේ හදන්න පුළුවන් microorganism means එයා host කෙනෙක් ගාවට ගියාම host ගේ cells අල්ලන් ඒවා attract කරගෙන ඒවට damage කරන්න පුළුවන් වෙන්න ඕනනේ. right. එන්න එහෙම කරන්න එයා ගාව තියෙනනේ විශේෂ ලක්ෂණ. දැන් හිතන්න. Now if we take a tiger, he is carnivorous. එහෙනම් carnivorous නම් එයා මුවෙක් වගේ ඉඳලා හරියනවද? නෑ නේද? එයාට තියෙනවනේ එක එක body features. It can run fast, good eyesight, then sharp teeth. ඒවා තියෙන්න ඕන නේද? නේද අයිෂා? මුවෙක් වගේ හිටියොත් එයාට පුළුවන් සතු මරන් ගන්න පුළුවන්ද? බෑ. ඉතින් ඒගොල්ලන්ට unique වෙච්ච characteristics. ඉතින් ඒගොල්ලන්ගේ වැඩේට හරියන characteristics ඒගොල්ලන්ට තියෙන්න ඕන. Right. First one, having optimal growth conditions that corresponds to the body conditions of the host. තේරුණාද? host ගේ body conditions වලට ගැලපෙන condition growth condition එකක් යාට තියෙන්න ඕනේ temperature එක ගත්තොත්. හරිද? හිතන්න දැන් එයාට ඉන්න බෑ කියලා දැන් ඉන්න host ජීවත් වෙන temperature එකේ microorganism ට ජීවත් වෙන්න බැයි නම් එයා වැඩෙන් grow වෙන්නේ නැත්තම් එයාට පුළුවන් ද host කරදර කරන්න බෑ. ඒක කරන්න බෑ. Having structures to adhere to the host cell and protect against host defense mechanism, capsule, villi. Then producing toxins. Toxins are then belong endotoxin and exotoxins. We are going to discuss about endotoxins and exotoxins later. Then having enzymes for invasiveness, such as phospholipase, vesitinase, hyaluronidase. And having enzymes such as DNase to alter the host metabolic process. Right? This kind of question asked in 2021 A-level structured paper. Right? So you have to study these 5 points. These are the 5 characteristics, right? Yes. Then next one, virulence and virulent factor, right? Virulence, it is the degree of pathogenicity of the pathogen. ලෙඩේ හදන්න පුළුවන්කම يعني pathogenicity, a degree of the pathogenicity. Now, if we take the chickenpox virus and if we compare the chickenpox virus with other viruses, this chickenpox virus, it is highly virulent, right? What are these virulent factors then? Virulent factors means the genes of the pathogenic microorganism that express factors which provide them the ability to infect their host and cause disease. First, microorganism, they express their pathogenicity. Pathogenicity means the ability of a pathogen to cause disease in the host by overcoming host defense mechanism. So this is called virulence. Virulence is the degree of pathogenicity of a pathogen. Some pathogens, they are highly virulent. Powerful, right? But some are non-virulent or less virulent. Then the relationship between this host and pathogen is a dynamic. Dynamic, it can change. Each modifies the activities and function of the other. Now look at this one, host and the pathogen. Pathogen has the pathogenicity. Pathogenicity කියන්නේ ලෙඩේ හදන්න පුළුවන් හැකියාව. host තියෙනවා host defense mechanism එක. හරිද? මේක තරාදියක් වගේ. host defense mechanism එකයි pathogenicity ගේ pathogenicity එකයි දෙක සමාන වුණොත් මොකද වෙන්නේ? ලෙඩේ හැදෙන්නේ ලෙඩේ හැදුනට ඒක එච්චර අපිට කරදරයක් නෙමෙයි. අපි ගත්තොත් අපිට කියලා වෛරස් කෙනෙක් වෛරල් ඩිසීස් එකක් කියන්නේ. හැබැයි host, right? If the host defense mechanism is very powerful and pathogenicity of the pathogen is very less powerful, then can they cause disease for us? No, they can't. අපිට ලෙඩේ හැදෙන්නේ නැහැ එතකොට. හැබැයි බැරි වෙලා හරි අර COVID-19 වගේ ගත්තොත් එයා කොහමද? එයා හරිම powerful, right? Virulence is very high, highly virulent. But the host defense mechanism, එයා weak. එතකොට මොකද වෙන්නේ? අර පැත්ත උඩ යනවා. එහෙනම් අපිට අනිවාර්යෙන් ලෙඩේ හැදෙනවා. අන්න ඒකයි මෙන්න මේ හින්දා තමයි මේක dynamic කියන්නේ. That is why this is called, this is a dynamic relationship. The relationship between a host and the pathogen is dynamic. Each modifies the activities and functions of the other. As a result, outcome of such a relationship depends on the virulence of the pathogen and the effectiveness of the host defense mechanism. Virulent factors enhance the pathogenicity and allows pathogen to invade and colonize host tissue and disrupt the normal body function, right? Here the pathogens, they use 2 major mechanisms for pathogenicity, invasiveness and toxigenicity, and we will discuss them in the next slide, right? So if you did something happen? Did you hear I explained this whole slide? I explained this whole slide. Did you hear? Aisha? No. Oh, yeah. Okay. Now, බලන්න, virulence and virulent factors. Microbes express their pathogenicity by means of their virulence. Virulence is the degree of pathogenicity of the pathogen. Some pathogens are highly virulent, whereas others are less virulent or non-virulent. And virulent factor means they are genes of pathogenic microorganisms which express factors which provide them the ability to infect their host and cause disease. The relationship between the host and pathogen is dynamic, right? And dynamic if the host defense mechanism and the pathogenicity of the pathogen, they are same, like taradil wage tiyenawa නම් balance එකේ තියනවා නම් හරිද? එතනදී ලෙඩේ හැදුනට it's not very harmful, right? The host defense mechanism can overcome it. But if the host defense mechanism is very powerful and the pathogenicity of the pathogen is like very low, what will happen? Can that pathogen cause the disease? No, it can't. But if we, for example, if we take COVID-19, the pathogenicity of the pathogen is very high and the host defense mechanism, it is very weak. So the pathogen can cause the disease. Okay, so that is why it is called dynamic, right? Dynamic. Each modifies the activities and functions of the other. As a result, outcome of such a relationship depends on the virulence of the pathogen and the effectiveness of the host defense mechanism. Virulence factor enhance the pathogenicity and allow the pathogen to invade and colonize the host tissue and disrupt the normal body function. Pathogens use two major mechanisms for pathogenicity, invasiveness and toxigenicity. Okay? If you need, you can copy down this one, right? And let me know. Finish? Harida? Aisha? Aisha and Nisma finish? Right. Okay. Then, now we are going to learn about those two mechanisms, right? First one, invasiveness. Here, the micropathogen, it invades the tissues. It invades the tissue of the host, right? It can overcome the host defense mechanism and invade the tissue and then multiply and colonize in the host, right? For that, they use some extracellular enzymes. They can produce some extracellular enzymes and they use those extracellular enzymes to invade the tissues of the host. First one is phospholipase. Phospholipase, it is an extracellular enzyme. It destroys animal cell membrane. Animal cell membrane එක destroy කරගෙන cells ඇතුළට යන්න පුළුවන්. Next one, lecithinase. Hydrolyze the lecithin component of the lipid in the cell membrane. And hyaluronidase destroy the body tissues by breaking down the hyaluronic acid, which is cementing substance in between cells. And the cells එකකට cement දාලා වගේ බැඳලා තියෙන cementing substance එක තමයි මේ hyaluronic acid එක කියන්නේ. මේගොල්ලෝ මොකද කරන්නේ? මේක කඩනවා. Right? කඩලා tissues destroy කරනවා. Right? Here, in this mechanism, the pathogen, they can produce extracellular enzymes and they invade the tissues by overcoming the host defense mechanism and multiply and colonizing the host and cause the disease, right? So these pathogenic microorganisms, they enter passively, right? through wounds on the skin, respiratory, gastrointestinal and genitourinary tracts. හරිද? තුවාල වලින් එන්න පුළුවන්, respiratory tract එකේ එන්න පුළුවන්, digestive tract එකේ එන්න පුළුවන්, genitourinary tracts වලින් එන්න පුළුවන්. Right. Next one. Toxigenicity. Right? Here toxigenicity means it is the second mechanism. Here the microorganism, they can produce toxins. Toxin, it is a biochemical substance. They can produce toxins, and those toxins, they can disrupt the normal function of cells. Biochemical substance එක toxins. හරිද? Toxins කියන එක જાතිය. ඒකෙන් මොකද වෙන්නේ? They can disrupt the normal functions of the cell. There are two types of toxins. These are biologically poisons, right? Endotoxins, and the second type is exotoxins. Now in the exam, they can ask you to compare endotoxins and exotoxins. So listen carefully. Endotoxins. Endotoxins are lipopolysaccharides. And exotoxins are they are proteins. Okay? Exotoxins are proteins. Endotoxins are lipopolysaccharides. Since endotoxins are lipopolysaccharides, they are thermostable. But exotoxins, they are proteins. So are they thermostable? No. Right? They are not thermostable. Right? They are thermolabile. Then endotoxins, they are they are a part of the microbial cell, and they are released when the bacteria die and the cell wall breaks apart. Endotoxin කියන්නේ bacteria cell එකක් ගත්තොත් එයාගේ endotoxins කියන්නේ එයාගේ part එකක්. එතකොට එයා මැරිලා cell wall එක break වුණාම තමයි එයා එළියට එන්නේ. හරිද? But these exotoxins, right, they are produced inside the bacterial cell as a part of their growth and metabolism, and those exotoxins are released to the surrounding environment after cell lysis. Right? Okay? So these in the exotoxins, the catalytic nature, due to their catalytic nature, even a small amount is very harmful. Right? Very harmful. And these exotoxins, they are most commonly produced by gram-positive bacteria and few gram-negative. You know, depending on the amount of peptidoglycan in the cell wall of bacteria, they can divide as gram-positive and gram-negative. So these exotoxins are produced by gram-positive bacteria and few gram-negative bacteria. But endotoxins, they are produced only by gram-negative bacteria. An example for endotoxin is the liposaccharides of the cell wall of Salmonella tymphi. Right? Yes. Toxigenicity. Ability of microorganisms to produce biochemical substances known as toxins that disrupt the normal function of cells. These are proteins or lipopolysaccharides that produce specific harmful effects on the host. Thus are called biological poisons. They may be endotoxins or exotoxins. Endotoxins බලන්න මේකෙන් S එකක් එන්නත් පුළුවන්. හරිද? Endotoxins are lipopolysaccharides. Right? It does say I told you exotoxins, they are proteins. These are thermostable toxins which are part of microbial cell. Toxins are released when the bacteria die and the cell wall breaks apart. All endotoxins cause the same signs of symptoms regardless of the species of pathogen. These symptoms include chills, fever, weakness, generalized itches, and sometimes shock and teeth. Endotoxins are produced only by gram-negative bacteria. Example, lipopolysaccharides of the cell wall of Salmonella tymphi. Then exotoxins. Exotoxins are produced inside bacterial cells as part of their growth and metabolism, and are secreted or released to the surrounding environment after cell lysis. Exotoxins are proteins. Majority of them are enzymes due to their catalytic nature. Even a small amount of toxin is quite harmful. These are thermolabile protein toxins being inactivated by boiling. Exotoxins are most commonly produced by gram-positive bacteria and few gram-negative bacteria, right? So, exotoxins are classified into three types: neurotoxins, enterotoxins, and cytotoxins. Neurotoxins interfere with the normal transmission of nerve impulses. Toxins produced by Clostridium tetani. Enterotoxin stimulates the cell of the gastrointestinal tract in an abnormal way. Example, toxins produced by Vibrio cholera. Cytotoxins kill the host cell by enzymatic attack. Example, toxin produced by Corynebacterium diphtheriae. Now, right. Important diseases of human caused by microorganism, right? You know, our whole body can affected by microorganism, and there are different portals of entries. It can be a virus, bacterial, right? It can be a viral disease or bacterial disease or fungi, right? Yes. First one, skin. Skin. Chickenpox. Chickenpox, it is a viral disease, right? The causal agent is herpes virus varicella zoster. Herpes virus varicella zoster. So normally, then herpes virus varicella zoster, you don't have to italicize or underline because it's not a scientific name, right? And now you can see like red patches on the skin. Then the rubella virus. Rubella, this is rubella, caused by rubella virus. And measles, right? Measles virus, measles virus. Here the portal of entry is respiratory tract. එතකොට මේ herpes virus, varicella zoster, rubella virus, measles virus, they all enter to our body through the respiratory tract and they affect the skin, right? They affect the skin. You can see in all those three diseases, there are red rashes on the skin, ne? Red rashes. Right. Then conjunctivitis, right? When the affected organ is eye. Now the disease is conjunctivitis. It can be a bacteria or viral one. Now this is a healthy eye. In the first picture, in the first diagram, they have shown a healthy eye. But this is a healthy eye. And this is a viral conjunctivitis. Viral conjunctivitis. And this is a bacterial conjunctivitis. And this is an allergic one. Allergic. Now compare. Now in the viral conjunctivitis, then here the amount of like the මෙතන නිකන් අර මොනවද නිකන් මෙතන ඇහැ ඉදිමිලා නේද? In bacterial conjunctivitis. Now here in the allergic conjunctivitis ඇහැ ඉදිමිලා වගේ පේනවා. මෙතන fluid formation එක bacterial වල අඩුයි. But here it is high. In allergic condition it's very high. Right? Anyway, you don't have to write down their differences. Anyway, conjunctivitis is caused by bacteria or virus, right? Haemophilus influenza, it is the bacteria that cause bacterial conjunctivitis. And adenovirus is the virus who cause viral conjunctivitis. Again, the portal of entry is respiratory tract. Bacterial meningitis. Meningitis. Now here the affected organ is the nervous system. Right? Bacterial meningitis. So this is caused by bacteria. Streptococcus pneumonia, the Haemophilus influenza, Neisseria meningitidis, they can cause bacterial meningitis. So here again the portal of entry is respiratory tract. Then skin. Then we have the skull, durameter, arachnoid meter, and the blood vessels. දන්නනේ පයාමීටර් ඇරක්නොයිඩ් මේක ඩුරාමීටර් right layers ටික ඊට පස්සේ pathogenic agents enter through the blood right so who are the causal agents Streptococcus pneumonia, Haemophilus influenza, Neisseria meningitidis. It's a bacterial disease. Then tetanus. Again, the affected organ is nervous system, right? Tetanus caused by bacteria, Clostridium tetani, right? Tetani. මලකඩ කාපු ඒවෙන් එහෙම තමයි එන්නේ. Tetanus, Clostridium tetani. So the portal of entry is damaged skin. Then rabies, ජලබීතිකාව, rabies, ලස්සන පින්තූරයක් තියෙනවා. Rabies virus, it's a viral disease, right? The wound on the skin. හරිද? කකුලේ හරි කොහේ හරි ඇඟේ තුවාලයක් තියලා ඒකේ බලු කෙල එහෙම මොන හරි ගෑවුණා අන්න rabies හැදෙන්න පුළුවන්. It's also not only by dogs. It can be caused by other mammals also, right? Baby rats, cows, okay? Then rheumatic fever. Cardiovascular system. It affects the cardiovascular system. Again, this is a bacteria caused by Streptococcus pyogenes. Streptococcus pyogenes, right? Right. Skin. Chickenpox caused by herpes virus varicella zoster. Right. You can include portal of entry here. Cause the causal agent they enter through the respiratory tract. Then rubella virus. Measles virus. Eye conjunctivitis. Bacteria or virus. Make Haemophilus kiyanne, please underline that one. Haemophilus influenzae. Haemophilus influenzae is one, right? So one causal agent. It's a bacteria. Haemophilus influenzae. Here Haemophilus also should be italicized. And adenovirus kiyanne මේක හදන virus ගේ නම. Nervous system. Bacterial meningitis, Streptococcus pneumonia, Haemophilus influenzae, Neisseria meningitidis. Right? Over දැම්මම පාඩම් කරන් ඉන්නේ exam එකට කලින් දැස්සේ ඔය පාඩම් කරන්න බෑ. හරිද? Nervous system, again tetanus. Tetanus කියන්නේ මොකද්ද? Bacteria එකක් හදන්නේ. Clostridium tetani. Write down bacteria caused by bacteria. Rabies, viral disease. Rabies virus. Then rheumatic fever caused by a bacteria. It affects the cardiovascular system, blood, right? Streptococcus pyogenes. Tuberculosis. It affects what? Respiratory system. Mycobacterium tuberculosis caused by a bacteria. Influenza virus, right? Influenza virus. Pneumonia, again bacteria, Streptococcus pneumoniae. Then digestive system, hepatitis, right? Hepatitis virus, hepatitis A virus. Food poisoning, Staphylococcus aureus. Then cholera, Vibrio cholera, right? Contaminate water එහෙම බිව්වම හරිද? Typhoid, Salmonella, tymphi. And Salmonella tymphi, Vibrio cholera, then Staphylococcus aureus. They all should be italicized. And may hepatitis A virus ඒක normal විදිහට ලියන්න ඕනේ because it's a virus. හරිද? ඒව හදාගන්න ඔයගොල්ලන්ගේ resource book එකේ. Urinary system, leptospirosis. Leptospirosis caused by a bacteria, Leptospira interrogans. Then reproductive system, gonorrhea, Neisseria gonorrhea. So they are sexually transmitted diseases. So it's affected organ is reproductive system. Genital herpes simplex virus. And the next one, AIDS. Then AIDS, you in the reproductive system, you learn AIDS under sexually transmitted disease. But it affects not to the reproductive system, it affects to the immune system. It's an MCQ point. හරිද? නිකන් පිස්සුවෙන් වගේ ගිහලා AIDS කිව්වහම ඒක reproductive system එකට affect කරනවා කියලා ගහන්න එපා. ඒක affect වෙන්නේ කාටද? immune system එක තමයි ආ attack කරන්නේ. immune system එක weak කරන එක තමයි ආ කරන්නේ. Human immune deficiency virus, HIV. Okay? හරිද? Any doubts? No doubts. No. The only doubt is how we can study this table, neither. But you have to study, right? MCQs වලට අහන්න පුළුවන්. Then, methods of controlling microbial diseases of human. Right. Control methods. දැන් microorganisms ලා ඉගෙන ගත්තා. ඒගොල්ලෝ හදන ලෙඩ ඉගෙන ගත්තා. දැන් ඉතින් මේවා අපි control කරන්නත් එපා. Right. First one. We can use antiseptics and disinfectants, right? Or else we can use antibiotics. If not, we can undergo immunization. Antiseptics, disinfectants පාවිච්චි කරන්න පුළුවන්, නැත්නම් antibiotics පාවිච්චි කරන්න පුළුවන්. එහෙම නැත්නම් vaccines use කරන්න පුළුවන්, control කරන්න. හරිද? Right. Antiseptics and disinfectants. දැන් මේ දෙක වෙනස්. Anyway, these are chemical substances that used to kill or reduce the microbial population in order to prevent infection. Right? Now, antiseptics, disinfectants, they are liquids. Right? Liquids. And their effectiveness, it varies with the concentration, the duration of exposure, temperature, and presence of the organic matter. The difference between antiseptic and disinfectant is that the antiseptic we can apply this antiseptic directly apply to the human body, but disinfectants we cannot apply to the human body. හරිද? So antiseptics, they are used to disinfect living surfaces as skin, ethanol, isopropanol, chloroxylenol. දැන් ඔය තුවාලයක් එහෙම සුද්ධ කරන්න මොනවද දාන්නේ? isopropanol, ethanol ඕවා use කරන්නේ නේද? නැතුව මේ අර টয়ලට් හොදන ෆීනෝල් ගෙනලා තුවාල හොද කරනවද? අයියෝ කොමඩත් මේව කරන්න හොඳයි ෆීනෝල්. ඉතින් isopropanol දානට වඩා ෆීනෝල් හොඳයි කියලා කිසිම කරනවද? නැහැ. Antiseptics and disinfectants both are liquids, formulated as liquids, and we can apply antiseptics directly to the human body. But disinfectants, they are used to disinfection of non-living surfaces. Okay? Antiseptics example, ethanol, propanol, isopropanol, chloroxylenol. Disinfectants, phenol, hypochlorites. හරිද? හරි නේද? වෙනස මතක තියාගන්න. Antiseptics and disinfectants are chemical substances that are used to kill or reduce microbial population in order to prevent infection. However, such chemicals are not effective against some microorganisms. For example, poliovirus, tuberculosis bacteria, spores of bacteria and fungi are not destroyed by most antiseptic and disinfectants. The major difference between antiseptics and disinfectants is that antiseptics can be safely and directly applied to the human body, whereas disinfectants cannot. Therefore, antiseptics are used in disinfections of living surfaces such as skin, right? Disinfectants are used in disinfection of non-living surfaces, such as operation theaters, then bathing areas, sink, kitchen tops, cutlery, drains, etc. Antiseptics and disinfectants are generally formulated as liquids. Their effectiveness varies with concentration, duration of exposure, temperature, and presence of organic matter. Some examples of antiseptics and disinfectants are given below. Antiseptics, ethanol, isopropanol, chloroxylenol. Disinfectants, phenol, hypochlorites, calcium hypochlorite and sodium hypochlorite. Then antibiotics. Right. Antibiotics controlling microbial diseases. When the body defense fails to protect body from the infection or overcome the disease, it has to be treated by chemotherapy with antimicrobial drugs. Antimicrobial drugs kill or interfere with the growth of microorganisms without damaging the host. Antibiotics are effective antimicrobial drugs against bacteria. Some antibiotics effect against broad range of bacteria and they are termed as broad-spectrum antibiotics, while others affect only against a specific group of bacteria and are called narrow-spectrum antibiotics. In antibiotics පාවිච්චි කරන්නේ මොන જાතියටද? බැක්ටීරියාට. එතකොට අපේ immune system එකෙන් ඒ ලෙඩේ overcome කරන්න බැරි නම් තමයි we have to use these antimicrobial drugs. තේරුණාද? right? ආසාවට antibiotics ගත්තාම මොකද වෙන්නේ? ඇත්තට ලෙඩක් හැදුනහම හොඳ කරගන්න බැරි වෙනවා. Right? So when using these antibiotics, you should be careful. හිතේ හැටියට අරන් බොන්න හොඳ නැහැ. පාවිච්චි කරන්න හොඳ නැහැ. So and there are two types as broad spectrum antibiotics and narrow spectrum antibiotics. A broad spectrum antibiotic means the antibiotics affect against a broad range of bacteria, and narrow spectrum antibiotic means the antibiotics that affect only against specific group of bacteria. Right? This is also, I think, like this is given in the 2021 A-level paper. Penicillin, inhibition of cell wall synthesis. Erythromycin and tetracycline, inhibition of protein synthesis. Daptomycin, disrupting plasma membrane. Rifampin, inhibition of DNA or RNA synthesis. Okay, then the third way to avoid and prevent microbial diseases. That is immunization of vaccine. Right. First of all, you should know the definition of vaccine. Vaccine is a suspension of weakened pathogen or fractions of organism that is used to induce the immunity. Then for the COVID-19, it's a virus, right? Now we use ethanol or ඔක්කොම use කරලා sanitizers ඒවා මොකද්ද කරේ? We use those things for disinfections as antiseptics, right? Also we use antibiotics, but we can't control this virus using those two methods. After that, right, what was our third thing? What is the third one? Third step? In all the countries, they vaccinated the people. They vaccinated the population. Vaccines dunna. COVID-19 vaccine dunna neda. Doses තුනක් කොහෙද දුන්නා. ඒ කියන්නේ ඒ virus එච්චරටම harmful. ඒව අර methods දෙකෙන් අපිට control කරන්න බැරි වුණා. Control උනේ මොකෙන්ද? vaccine එකෙන්. තේරුණාද? vaccine එකෙන් තමයි ආව control කරගත්තේ. So, vaccines, they are frequently used to control diseases caused by viruses. The virus වලින් හදන disease control කරන්න තමයි අපි vaccine පාවිච්චි කරන්නේ. because antibiotics තියෙන්නේ bacteria වලට, ඊට පස්සේ අර antiseptics, disinfectants වලින් මේවා control කරන්න බෑ. Right? අඩු කරන්න පුළුවන්, but we can't it is not a very good way. There are several types of vaccines. There are three types: live attenuated vaccine, inactivated vaccine, and subunit vaccine. Okay, right. Live attenuated means live, right? From the word you can get the definition. Live attenuated means this vaccine, it contains the live pathogen which is weakened for its pathogenicity. Deliberately weakened. The word is very important. Deliberately weakened. Vaccine එක ඇතුළේ ඉන්නවා live pathogen. හැබැයි එයා මොකද weak කරලා තියෙන්නේ. එයාගේ දුර්වල කරලා weak කරලා එයාගේ pathogenicity එක අඩු කරලා තියෙන්නේ. හරිද? එහෙනම් මේ vaccine එක අපි ගහගත්තා කියන්නේ අපිට අනිවාර්යෙන් අර නිකන් ලෙඩේ හැදුනා වගේ එකක් තමයි වෙන්නේ. හරිද? So it is inside, active inside our body. Okay? So since that pathogen is active, our immune system will produce, right? Our immune system produce it try to protect our body from this host. හරිද? එයාව control කරන්න අවශ්‍යව අපේ ඇඟ ඇතුලේ හදනවා. හැබැයි එතකොට ඇත්තටම ලෙඩක් හැදුනොත් මොකද වෙන්නේ? එහෙනම් අපේ බොඩි එක රෙඩි පිට තමයි ඉන්නේ. එයාට ඇත්තටම මේ ලෙඩේ හැදුනොත් රෙඩි පිට ඉන්නේ. right? එතකොට අර control කරන්න පුළුවන්. So here we don't need any secondary immunization. ආයි පාරක් තව dosage එකක් ගන්න උවමනාවක් වෙන්නේ නැහැ. Vaccine contain live pathogens which were deliberately weakened for its pathogenicity. දැන් ඔය MMR, measles, mumps and rubella. ඔය පොඩි කාලෙම ගහනව නේද? වයස ගියද ගහන්න කියලා මතක නැහැ. ඉස්කෝලේටත් ඇවිල්ලා ඔයගලන්ට ගහනවා ඇති ඔය vaccine. හරිද? එකපාරයි ගහන්නේ, ආයි ගහන්න ඕනේ නැහැ. It gives lifelong immunity. Vaccine contain live pathogens which were deliberately weakened for its pathogenicity. These vaccines mimic an actual infection. Since the pathogen is active inside the host, such vaccines provide lifelong immunity. More often, a booster immunization is not required. Examples for live attenuated vaccine are vaccines for measles, mumps and rubella, and chickenpox. Right? The second group is inactivated vaccine. Inactivated vaccine means the pathogenic microorganism, it is inactivated or killed in the vaccine. Now here it's a live pathogen is used, but in the inactivated vaccine, the microorganism, it is inactivated or killed. Right? Here, this inactivated pathogen vaccine, we need to have repeated booster doses. We can boosters, we can එකපාරක් කරන් මදි දෙතුන් පාරක් වුණ ගන්න වෙනවා. හරිද? So this is given for viral diseases such as rabies. Then rabies වලට එහෙම නේද? ඔයගොල්ලෝ නිකන් බල්ලෙක් පූසෙක් කෑවොත් මොකද අම්මලා කරන්නේ? අරගෙන යනවා ડોක්ටර් ගාවට. ගහනවා දෙකක් නෑ. දවස් කිහිපයක් එන්න කියනවා හතක්ද කොහෙද ගහනවා නේද? ඩෝසස් හතක් දෙනවා ඔයගලන්ට. නේද? right? So rabies, influenza, polio වලට තමයි දෙන්නේ viral diseases. Then bacterial diseases such as cholera. Right? Inactivated vaccine. Pathogenic microorganism is inactivated or killed in the vaccine. Compared to live attenuated vaccine, inactivated killed vaccines often require repeated booster doses. Examples for inactivated vaccines are vaccines for virus diseases such as rabies, influenza, and polio, and bacterial diseases such as cholera. And the last type is subunit. Now, this subunit vaccine, they do not contain the whole microorganism. They contain only the antigenic fragments or the toxins. එයාගේ toxins හරි antigenic fragments තමයි මෙයාගේ තියෙන්නේ. So when we get these vaccines, what happens? These antigenic fragments, now they are present in our body. So it induces the immunity. It induces our immunity system, right? So the best example for a toxoid vaccine, that means that subunit vaccine, it contains toxin of the pathogenic microorganism, right? So example is the vaccine given for tetanus and diphtheria. And also now in the present, now in these days, subunit vaccines are also produced by genetic engineering. The example is the vaccine given for the hepatitis B vaccine. But these vaccines, they usually require repeated booster doses to obtain the full immunity. Right. Subunit vaccines contain only the antigenic fragments of a pathogen that can induce immunity in the recipient. Toxoid vaccines are the best example for subunit vaccines. Those have been used for a long time. Toxoid contain inactivated toxins derived from a pathogen. Examples for toxoid vaccine are vaccines for tetanus, diphtheria, etc. Presently, subunit vaccines are produced using genetic engineering. Example, hepatitis B vaccine. Subunit vaccines usually require repeated booster dose to obtain full immunity. Okay? Hari neda? Now I'm going to start the third section of today's session. That is, use of microorganisms in industry. Any doubts? Right. All right. Use of microorganism in industry, agriculture, and environment. Microorganisms have been exploited for various purposes long before their discovery. In the Babylonian and Sumerian civilization, they used yeast to make alcohol. Right? But after the discovery of organisms, microorganisms, right, not organisms, microorganisms, in the later part of the 19th century, pure cultures of microorganisms are being used in the food production. This increased the understanding of microorganisms, their processes and products. At present, various industries based on selected microorganisms and their qualities are in operation. That. So, like for the industries, normally chemical processes are used. But instead of these chemical processes, we can use microbial processes. Right? What are the advantages of using microbial processes over chemical process? Now, if we take a chemical process, we have to give high pressures, high temperatures, right? Then, so the production cost is very high. But if we take microbes, right? Their nutritional requirements are very simple, and they can convert a wide range of raw materials, right? They can convert cheap raw materials into industrially important products, right? Then අපි ගත්තොත් alcoholic beverages, wine, arrack, නේ? උවහදාන්නේ මොනවද නිකන් අර ඔය එක එක මොනවද? Sucrose solutions, right? Or තමයි fermentation කරලා තමයි ඔක හදන්නේ. Right? Yes. Then due to high growth rate, they can convert the raw material into product within a short period of time. The average generation time of microorganism is relatively low. So during a short period of time, they can convert the raw materials into products. And their growth conditions can be controlled to obtain desired end products. Then, now in chemistry also you may have learned this part. Alcohol, vinegar, ethanol production. මොකද්ද කරන්නේ? ඉස්සෙල්ලාම දැන් අපි ගත්තොත් sugar solution එකක් තියෙනවා. First දැන් අර අපි chemistry වලින් කියලා දෙමු. දැන් පොල් රා. හරි පොල් ගහේ. ඒ පොල් මල තියෙනවානේ ලොකු මල දැකලා තියෙනවා නේද? නිකන් අර ඇත්දලේ වගේ තියෙන්නේ. දන්නවා නේද? චූටි මල නෙමෙයි ලොකු මල. right? Inflorescence කියන්නේ අර ලොකු මල තියෙන ඒක අරන් මොකද කරන්නේ? ඒක අර අග්ගිස්ස කපරනේ. කපලා ඔකට මුට්ටියක් යට ගහනවානේ. යට ගහලා තියෙන ඊට පස්සේ මොකද වෙන්නේ? අරකේ තියෙන පැණි ටික ඔක්කොම අරකට බේරෙනවා. හරිද? ඊට පස්සේ වටෙන් යන කවුරු හරි ඊස්ලා හිටියොත් මොකද වෙන්නේ? අර මුට්ටිය ඇතුළට බහැලා ඒගොල්ලෝ මොකද කරන්නේ? They secrete sucrase. Right? Now the sucrose, when due to the action of these sucrase, it is converted into, right? Due to different enzymes secreted by this yeast, it converts into ethanol. හරිද? ඕක අපහු purify කරලා ගත්තාම මොකද වෙන්නේ? Okay. Purify කරලා ඕක තව තිබ්බොත් මොකද වෙන්නේ? ආපහු ඕක oxidize වෙනවා. Right? Acetic acid bacteria වගේ ආවම ඕක ආයි carboxylic acid වෙනකම්ම oxidize වෙනවා. Acetic acid එකක් විදිහටම oxidize වෙනවා. නේද? එතකොට ඒක අපිට මොකක් විදිහටද ගන්න පුළුවන්? විනාකිරි විදිහට ගන්න පුළුවන්. එතකොට මෙන්න ඒකම their growth condition can be controlled to obtain desired end products. එතකොට අපි අතනදී අර if we stop the further oxidation of ethanol, right? We can obtain like ethanolic alcoholic products. Right? But if we let it to oxidize further, then we can obtain vinegar. Right? Reactions can be carried out at low temperatures, energy and pressure compared to the conventional industrial methods. They give higher yield with a higher specificity when compared to the conventional industrial methods. Microbes are amendable to genetic manipulation to obtain desired yield and quality with high efficiency. Right. So those are the advantages of using microbial processes over chemical processes. Then basic principles of metabolic processes of microorganisms for product formation. First one, microbial cells used as end product. Single cell protein, yeast, cholera, right? Then microbial metabolic products are used as end products. Primary end products වෙන්න පුළුවන්, secondary metabolites වෙන්න පුළුවන්. Then alcoholic beverage කියන්නේ මොකද්ද? Primary end product එක. Sucrose is converted into ethanol. Right? Secondary metabolites, antibiotics. If not, microbial metabolic processes are used as end product. Bioremediation, metal extraction, directing. Fourth one, genetically modified microorganisms are used to produce end products. Right? Production of commercial enzymes, vaccines, right? Subunit vaccine, like toxoid vaccine, hepatitis B virus, and hormones. Yes, application of microorganisms in industries. Industrial microbiology. Industrial microbiology is the large-scale production of economically important products using microorganisms and their metabolic processes. Right? These microorganisms, they are known as miniature chemical factories. Right? Because inside this microorganism, various energy releasing and energy acquiring chemical reactions takes place. Miniature chemical factories. So within those miniature chemical factories, raw materials or the substrate converted into the end products, right? And one or more byproduct and waste. So end products can be separated from the byproduct and waste by purification, right? To obtain purified industrial products. Right. Commercial products made by microorganisms and their processes. First one, single cell protein. The single cell protein, they are growing large scale as a food supplement. Then in a human population එක වැඩි වෙනවා. ඉතින් ඔය ඉන්න හැම කෙනාටම කන්න දෙන්න බෑ. නේද? ඒකයි ඕගොල්ලෝ වැඩි ඕගලන්ට කන්න දෙන්න බෑ කියලා එහෙම ඉන්න පුළුවන්ද බෑ. ඉන්න ලෝකේ ඉන්න මිනිස්සු ඉන්නවා හැමෝටම කන්න දෙන්න ඕන. හරිද? එතකොට ඒකට රෙමඩි එකක් තමයි මේ තියෙන්නේ. Microorganisms, they are like can be used as a food supplement, rich in proteins, yeast, spirulina species, chlorella species. Okay. Okay. When this one, this chemical reaction, it should go to the second one. හරිද? alcoholic and alcoholic beverages කියන තැනට තමයි මේක එන්නේ. මේ reaction එක මෙතන තියෙනවා නම් ඒක කපන්න resource එකේ. හරිද? ඒක මෙතනට අදාළ එකක් නෙමෙයි. දැන් ඔයගොල්ලෝ ගත්තොත් මොනවද කන්නේ? ඔයගොල්ලෝ අර marmite කන්නේ. marmite වල මොනවද තියෙන්නේ? yeast. It's a food supplement rich in new proteins, right? Alcohol and alcoholic beverages. Microorganisms are involved in the production of almost all alcoholic beverages such as beer, wine, sake, toddy, ethanol. Yeast, Saccharomyces cerevisiae, ferment sugars into ethanol and carbon dioxide. දැන් මේ මෙතන තියෙන reaction එකේ කතා කරන්නේ. Yeast උයි Saccharomyces cerevisiae මොකද කරන්නේ? Sugars fermentation කරනවා. As a result, they produce this ethanol plus carbon dioxide, right? Globally, දැන් මෙතන මොකද්ද පාවිච්චි කරලා තියෙන principle එක? They use microbial metabolic process. Globally, more than 70% of ethanol is produced by fermentation. Sucrose derived from sugarcane is the most widely used fermentation substrate. In addition, simple sugars derived from plant and dairy waste are also used. Then beer හදන්නේ මොනවද? By fermentation of cereal grain malt. හරිද? හාල්. Wine produced by fermentation of grapes or other suitable fruits. Toddy, rara, produced by fermentation of sap of palms such as palm oil and coconut. And arak, produced by fermentation of palm tree sap such as coconut and sugar cane. Right? ඔයගොල්ලන්ගේ මොනවාද chemistry පොතේ එහෙම තියෙනවද නේද? අරක්කු කියන්නේ බොහොම භයානක එකක්. අරක්කු හදන ක්‍රමයේ මෙන්න මේ ආකාරයෙන් කියලා තියෙනව නේද? තියෙනවද? දැකලා තියෙනව නේද? ඉන්ඩස්ට්‍රියල් වල තියෙන්නේ. Unit 40. Right. Production of vinegar, vinakiri. Again, microbial metabolic process is used as the principle. Right? So there are two steps in vinegar production. First one, alcoholic fermentation. Again, by the action of yeast or Saccharomyces cerevisiae, sugar is fermented into ethanol plus carbon dioxide. Then that ethanol is further fermented, right? That means it gets oxidized by the action of acetic acid bacteria and converted into acetic acid. අර චූටි කාලේ ඉගෙන ගන්න vinegar වල තියෙන මොකක්ද acid එක? weak acid, acetic acid. But alcoholic fermentation, sugars in malted grain, sap of palms, sugarcane and fruit juice is fermented by Saccharomyces cerevisiae. Ethanol is subjected to acetic acid fermentation. Acetic acid fermentation, ethanol derived from alcoholic fermentation undergoes incomplete oxidation and is converted into acetic acid. This process is highly aerobic and involves Acinetobacter species and Gluconobacter species, right? කාවද ගන්න Acinetobacter and Gluconobacter for the second step. හරිද? එතකොට මෙතන බලන්න in the first step they have used yeast, fungi කෙනෙක්. In the second step they have used bacteria, Acinetobacter and Gluconobacter. හරිද? Now here another difference. Alcoholic fermentation. This is an aerobic thing. Anaerobic එකක්. Anaerobic respiration takes place here. Here, aerobic respiration takes place. Highly aerobic. හරිද? Fermentation කියලා තිබ්බට මොකද්ද මේක? Aerobic. First step is anaerobic. Second step is anaerobic. First one anaerobic, ඒ කියන්නේ fermentation. Second one aerobic. හරිද? හරිද? ඒවා කෙලින්ම ඩිරෙක්ට්ලි දීලා නැති ඒවා ඔයගොල්ලෝ ඉතින් කියවද්දි ඒවා තේරුම් ගන්න ඕනේ. MCQ වල අහන්න පුළුවන්. මේ තියෙන්නේ එහෙම එකක්. Alcoholic fermentation, then we can get ethanol. කාවද use කරන්නේ? Yeast. Then it undergoes again when ethanol oxidizes, it produces acidic fermentation. This is done by acetic acid bacteria. And you get vinegar. Right. Dairy products. So, dairy products are made by the fermentation of milk. Sugar, lactose in milk is fermented by lactic acid and lactic acid producing bacteria into lactic acid. These bacteria are killed during pasteurization. therefore added externally when making dairy products. Curd yogurt is produced by fermentation of lactose sugar in milk by mixed population of Lactobacillus bulgaricus and Lactococcus lactis. Right? Lactis කියන එක මොකද්ද වෙන්න ඕනි? It should be italicized. Then S. thermophilus. Lactobacillus bulgaricus add flavor and Streptococcus species add creamy texture and flavor. Production of cheese, Streptococcus species and Penicillium molds. Lactic acid is commercially produced by using waste products from cheese and butter industry. Lactobacillus bulgaricus ferment lactose into lactic acid. Then organic acid. So organic acid can be obtained by microbial fermentation. එතකොට මෙතන use කරන substrate තමයි wheat, cane molasses, මොනවද cook තියෙන්නේ. Then organisms such as Aspergillus niger are used. Right? Aspergillus niger is a fungi. Right? Citric acid ගහදන්නේ කොහමද? Sucrose ferment කරනවා Aspergillus niger. Aspergillus niger කියන්නේ yeast කෙනෙක්. Right? And produce citric acid. Then metal extraction. Some metals from the ore are extracted with the help of microorganisms. What is the principle used here? Microbial metabolic processes. This process is called leaching. One of the best examples is extraction of copper from lower grades of copper ore, for which other extraction methods are unprofitable. Theobacillus peroxidans, again it should be underlined. Theobacillus peroxidans recover copper from the ore that contains iron and sulfur. Iron pyrites, FeS. About 70% of copper in the ore can be recovered by this microbial process. Uranium, gold, and cobalt ore are also leached in similar manner using microbial process. Right? So you should know what is leaching and the type of microorganism used to recover copper from the iron pyrites. Here iron and sulfur, O18 part gun. Where copper leaching? අර ඔයගොල්ලෝ ඉන්ඩස්ට්‍රියල් කෙමිස්ට්‍රි වල සල්ෆියුරික් ඇසිඩ් ප්‍රොඩක්ෂන් එකේදී ඉගෙන ගන්න ඇති. අයන් ඕ වල එහෙම වලින් සල්ෆර් ගනිද්දි මොකද වෙන්නේ? කෙලිම සල්ෆර් ගන්න ඕනේ SO2 අරගන්න පුළුවන්. නේද? SO2 ඔක්සිඩයිස් කරලා SO3 හදා ඊට පස්සේ සල්ෆියුරික් ඇසිඩ් හදන්න පුළුවන් කියලා එහෙමත් ඉගෙන ගන්නවා ඇති. Right? That is chemistry. Okay. Production of vitamins. Microbes can provide a source of an inexpensive source of vitamins for individual food supplement. හරිද? මේක විටමින් එක දීලා කවුද මේවා හදන්නේ? එහෙම අහන්න පුළුවන්. එහෙනම් විටමින් B12 හදන්නේ කවුද? Pseudomonas species, Propionibacterium species. Riboflavin, fermentation by fungi. And vitamin C කියන්නේ Acetobacter species. So, they are genetically modified microorganisms. That is the principle. Right? Vaccines. Right? Arapi ganagate subunit vaccines. Genetically modified. Right? Commercial production of vaccine is done by variety of microbial antigens used in active immunization against various diseases. Some of them are genetically engineered vaccine. Hepatitis B vaccine. Commercial production of various antibody preparation used for passive immunization. Right? You know, under immunity you learn active immunization and passive immunization. So for passive immunization, example, antitoxin against tetanus, right? Botulism toxin and immunoglobulin against rabies. Then enzymes. Again, microbial metabolic processes used. Amylase, Aspergillus niger, A. oryzae, Bacillus subtilis. Protease, A. oryzae, lipase, Rhizopus species, invertase, Saccharomyces cerevisiae, cellulase, A. niger. Again, Aspergillus niger. Aspergillus niger හදනවා amylase and cellulase. Aoricea හදනවා amylase protease. හරිද? Right. Then antibiotics. Right? Antibiotics, secondary metabolite of microorganism. Either used produced by microbial fermentation. The tetracycline, penicillin, streptomycin අපි කලින් ඉගෙන ගත්තේ. The tetracycline is produced by S. aureus fascians and penicillin by Penicillium chrysogenum. It is a fungi, and streptomycin by Streptomyces griseus. So, I think tetracycline, S. aureus fascians, penicillin, Penicillium chrysogenum, streptomycin, Streptomyces griseus. This is penicillium, it is a fungi, right? Okay. Then hormones. Genetically modified microorganisms. Human insulin. Human insulin is extracted from animal pancreas. Animal pancreas again insulin ගන්න එක it's a very expensive, right? එතකොට අවශ්‍ය ප්‍රමාණය demand එක වැඩි, right? So now at present insulin is produced chiefly by using genetically modified E. coli and Saccharomyces cerevisiae. And this insulin is identical to human insulin. Right? Insulin can more the diabetes than any other. Insulin, it can convert glucose into glycogen. Right? Then human growth hormone. Human growth in earlier is derived from horses, right? Animal derived hormones. But now it is produced genetically engineering by E. coli in large scale. Retin. ඕක අර මොනවද මේ දැකලා ඇති ඔයගොල්ලෝ. පොල් ලෙලි හැම පල් කරන්නේ නේද? පොල් ලෙලි සාමාන්‍ය පොල් ලෙල්ලක තියෙන පොල් කෙදි ගලවන එක ලේසි වැඩක්ද අමාරු වැඩක්ද? ඒක අමාරු වැඩක්. right? ඒක ලේසි වැඩක් නෙමෙයි. එතකොට දැන් ඔය කඹයම හදන්න මොකද කරන්නේ? පොල් ලෙලිනේ පාවිච්චි කරන්නේ. එතකොට ඉතින් පොල් ලෙල්ලෙන් කෙදි ටික අයින් කරගන්න එපා. එතකොට මොකද කරන්නේ? පොල් ලෙලි ලොකු වතුර වලවල් වල දාලා පල් කරනවා. හරිද? පල් කරද්දි මොකද වෙන්නේ? අර ඒව ඇතුලේ තියෙන නිකන් පේස්ට් එකක් වගේ එකක් තියෙනවා. ඕකේ බැක්ටීරියා ඇඩ් කරලා ඔය පේස්ට් එක මොකද වෙන්නේ? ඒගොල්ලෝ එක එක එන්සයිම්ස් දැම්මහම ඒ පේස්ට් එක ඒක අයින් වෙනවා. ඒක ලූස් වෙනවා. ෆයිබර්ස් ලූස් වෙනවා. ඒක තමයි එතන වෙන්නේ. Retin is the process of loosening fibers from woody stem or other plant materials such as coir. Ferretin plant materials are immersed in water for varying periods of time depending on the plant material. Heterogeneous bacterial population. Heterogeneous. Right? The jati godak etana. වෙනම නම් කියන බෑ. Heterogeneous bacterial population එක participate in the process under aerobic or anaerobic conditions. Bacteria secrete enzyme mainly pectinase. අර දන්නවනේ cell wall එකේ pectin තියෙනව නේද? carbohydrate එකක් විදිහට ඉගෙන ගත්ත pectin. pectinase දානවා to facilitate loosening. Next one, biogas production. Right? අර ගිය කාලේ හොඳට biogas ගැන කතා වුණේ gas නැති කාලේ මතකද? Anaerobic digestion of organic waste produce various gases called biogas. The type of biogas produced depend on the substrate biodegraded. Activity of acetogenic bacteria on the organic waste to produce carbon dioxide and hydrogen, whereas activity of methagenic bacteria produce methane. Acetogenic bacteria means it is a specialized group of anaerobic bacteria. They can generate acetate. Right. Next one, biofuel production. Now, petroleum කියන්නේ මොකද්ද? It's very expensive. හරිද? දැන් කියන්නේ අවුරුදු මෙච්චර එකක් යද්දි petroleum තියෙන ඒවා ඉවර වෙනවා කියලා. And I think ඒවා ඉවර වුණාම වාහන වතුර ගහලා දුවන්නද? බෑනේ. Right? There should be a replacement. එතකොට දැන් බ්‍රසීල් වගේ countries වල මොකද කරන්නේ? sugar cane එක කියලා use කරලා ethanol හදනවා. Right? Petroleum-based fuel supply is expensive and sometimes uncertain. As a result, much attention has been given to renewable replacement fuels such as ethanol, butanol, biodiesel, and biogas, etc. In Brazil, large amount of ethanol is produced by microbial fermentation of sugarcane to be used as a source for fuel. Also, efforts have been made to produce ethanol and butanol from cellulosic materials such as wood, waste paper, and corn stalk. දැන් අර දන්නවනේ ඇමරිකාවෙන් දැන් ඔය තිරිගු එහෙම අපිට පාම් පිටි ගේන්නේ, right? ගෙනල්ලා ඒව තියෙනනේ ස්ටෝක් එක පොත්ත තියනවානේ. පොත්ත අපිට දෙන්නේ නැහැ. ඒගොල්ලෝ පිටි ටික දීලා පොතු ටික අරගෙන යනවා. පොතු ටිකෙන් මොනවද කරන්නේ? මේ for the biofuel production, right? ඒක තමයි වටිනම එක ඒක අරගෙන යනවා. Corn stalks by using genetically modified bacteria. Many researchers are going on to produce biodiesel from microalgae. Sugars in bread, then bakery product. Sugars in bread dough is fermented by Saccharomyces cerevisiae. Then පාන් ගෙඩියක් යතින් පාන් පිටි ගුලියක් වගේ කනවා වගේ වැඩක් නෙමෙයි වෙන්නේ. පාන් ඇතුළ හරිම soft. නේද? ඇතුළේ හරි හිල් තියෙනවා ඒකේ පාන් ගෙඩිය ඇතුළේ. ඇයි ඒ? දැන් පිටි මෝලියා නේද මොකද කරන්නේ ඔයගොල්ලන් ගෙදර පාන් හදුවා කියලා හිතමු. ඊස්ට් දානවා. නේද? ඊස්ට් කියන්නේ Saccharomyces cerevisiae. හරිද? මේක මොකද කරන්නේ? ඒක ඇතුලේ තියෙනවානේ දැන් පාන් පිටි. පාන් පිටි කියන්නේ it's a carbohydrate. right? sucrose තියෙනවා. sucrose ටිකක් අරගෙන මොකද මේගොල්ලෝ කරන්නේ? ඒක fermentation කරනවා. කියන්නේ sucrose ටිකක් බිඳිලා ගිහිල්ලා මේ ඒ කියන්නේ starch ටිකක් බිඳිලා ගිහිල්ලා sucrose ඒවා හදනවා. හරිද? එතනින් ඊට පස්සේ දැන් sucrose තියෙනවා. sucrase වලට yeast sucrase release කරලා sucrase is converted into ethanol plus carbon dioxide. අර පාන් වලින් එනවා නේද නිකන් අර ethanol සුවඳ එන්නේ. අන්න ඒ හැදෙන ethanol නිසා. හරිද? එතන හැදෙනවානේ carbon dioxide. අර carbon dioxide හැදුනම මේක කියන්නේ ඒක gas එකක්. දැන් පිටි මෝලිය ඇතුලේ යාට යන්න ඉඩ නැහැ. ඉඩ නැති වුණාම මොකද වෙන්නේ? අර පිටි මෝලිය පිපෙනවා. තේරුණාද? පිටි මෝලියක් පිපෙනවා කියන්නේ ඔන්න ඔය carbon dioxide පිරෙන එක. ඒක පිටි මෝලියක් පිපෙනවා කියන එකෙන් අපි දන්නවා එහෙනම් මේ yeast වැඩ කරලා තියෙනවා කියලා. right? Sugars in bread dough is fermented by Saccharomyces cerevisiae, baker's yeast. The primary function of fermentation in bread is to generate carbon dioxide. Bread dough is made with grain flour such as wheat, rye, and rice. Dough traps carbon dioxide and rises due to pressure during baking and form open crumb texture. හරිද? ඉවරයි. හරි නේද? Any doubts? So that is the application of microorganisms in industry. And I'll upload I'll upload a worksheet from this part. Some essays, so you have to do it at home and bring to the class next day. Any doubts? Any doubts?